Thank you very much for the kind introduction. I would also like to thank the organizers for inviting me here to speak at this wonderful event. Thought I will take you through some of the efforts we've been doing in the... Can you hear me? In the lab over a number of years now, trying to model human neurodegenerative diseases, in particular Parkinson's disease, using induced pluripotency or iPSC technology. Before I do that, as a way of context on where and how we have been doing these activities, we're here, just close to Portugal, and this is the westmost part. Exactly. No, I say Spain is close to Portugal, of course. We are like the backyard of Portugal. And I was saying this is the westmost part of Europe, also the southmost part of Europe, and we're usually referred to as Southern Europe. All this story began at the turn of the century when, that's been mentioned, the first lines of human embryonic stem cells were derived in James Thomson's lab using human embryos, human blastocysts that were left over from IVF treatments. And we realized that we could not do that in Spain, and we could not do that in many European countries other than the UK. And I bring this up because there was a very active social movement instigated by scientists, and these are some of the, I don't know if you can read, but these are headlines from one of the main Spanish newspapers, and I cannot even read it myself, but that's probably 2000, 2001 when all this movement started, and we were basically complaining that we could not do that type of experiments in Spain. Scientists protest. That goes on until the political party that was in government at the time, which is a conservative party, decided that those experiments could not be done. We could not use human embryos for research. In the end, they caved, and this is 2003, when the government came out public saying that they were going to allow this. In the end, that type of social movement made the government to change in Spain on the basis of their response to this very problem. The change was introduced in 2003 in the law that regulated assisted reproduction technologies, introducing the possibility for leftover embryos that were discarded to be used for research, and there was a royal decree in 2004 regulating how these embryos and how this material could be used for research. And in summary, the mechanism that was adopted was this commission. There's a commission based in Madrid, a central commission for all Spain that is called the Commission on Guarantees for the Donation and Use of Human Materials and Cells. And this commission oversees all the projects that want to use embryos or similar type of research in Spain, and there's a centralized national bank for stem cells that is distributed in 3 nodes. One is in Barcelona, in my center, another one is in Valencia, and another one is in Granada, in the center of Al-Andalus. The way this system works is very simple, and I think can be also made as an example. It's been mentioned earlier, but it's really very simple. There's just a set of general principles, which are the samples have to be consented for donation after informed consent. The entire procedure has to be altruistic, and that raises some questions. It has to be scientifically justified, very general principles, and some red lines. So these are the only red lines that the Spanish legislation considers. Human embryos cannot be generated for the sole purpose of using them on research. We can not do reproductive cloning, and we cannot go beyond the 14-day limit that has been already mentioned. With those principles and red lines in mind, all the projects are submitted to this commission, and the commission authorizes or not the project. This is, as I said, a very simple project, a very simple process, a very simple system that has some advantages. One is that it is project-based, so some things can be good or scientifically or approved for one project and not for other project. It covers anything, so it has no limitations on what to cover. Anything, technology present or future, that uses embryos or that generates pluripotent cells, human, is covered. So it can adapt to future development, but it has, of course, some disadvantages. One of them, again, is project-based, so all the projects need to be submitted to this commission, and that is a time-consuming procedure. It can take up between 6 to 9 months to get your project approved, and it is very embryocentric in the sense that it didn't anticipate that 2 years later there was the discovery by the lab of Yamanaka saying that there was other way to make human pluripotent, well, in this case mouse, a year later human pluripotent cells that didn't require the destruction of an embryo. In any case, since we are generating human pluripotent stem cells, we are bound to use the same legislation. This technology, I think, has 3 main uses. There are hundreds of laboratories around the world using this type of technology, and there are 3 main uses. One is to understand the process of reprogramming itself, and it has provided very interesting insights into this phenomenon, into this process. The second use will be to generate cells for regenerative medicine, to treat conditions such as the one that have been discussed this morning, but I will center today the talk on the use of iPS cell technology to model human disease. And there are hundreds of diseases that have already been successfully modeled using this type of approach. Most of them are genetic conditions, monogenic diseases of early onset or congenital. And we wonder if this technology will also be suitable to model more complex diseases, diseases of the elderly, diseases associated to aging, or diseases with complex genetic bases. And for this purpose, we chose Parkinson's disease. Very summary, we were interested in this disease because it's a disease of aging, and the disease takes decades to evolve in the patient since the disease begins until the neurons die, and that was a challenge to see if we can reproduce something that in nature takes decades to occur to the timeline of an experiment of weeks, months in the lab. In the patients, what we see is 2 types of phenotypes. One, there is a specific type of neurons that die, and the neurons that don't die present, this type of neurons that don't die present accumulations that are known as Lewy bodies that contain aggregates of many proteins, but mostly alpha-synuclein. The other fact that was interesting about this disease is that like many other highly prevalent diseases, most of the cases are sporadic, but in some of them there is a familial history, and when you look at those families, you see that there are mutations that run in the family and that are associated to suffering the disease. In the case of Parkinson, there is a number of genes that have been identified, and I will just like to point your attention to this one, LRRK2. This is the most frequent cause of genetic or familial Parkinson's disease. So when we attempt to model Parkinson's or any other disease, there are very simple models. Well, of course, there's in here, I'm trying to plot the genetic complexity of the disease or the model against the organismal or cell complexity of your model. The patient of course, is not a model of the disease. I mean, the patient is a patient, but if you take an enough number of patients, you can have an idea of the disease, and it shows, of course, all the genetic complexity of the patient and all the genetic complexity of a living organism, a living human being. Simple models, such as here, these are cell-free models in which you check the effect of a mutated gene, the protein that it encodes, how that affects the activity or the efficiency of the protein. You can check that, and you have a very powerful information about what that protein or that regulatory network does. But in the absence of any complexity given by the cell environment or in the absence of other genes or other proteins. You can use cell-free systems to model 2, 3, 4, whatever number of genes you want, and you will have information about all those proteins or that network of proteins in the absence of cellular complexity. You can move, and this has all been used for modeling and studying Parkinson's disease. You can use systems in which you use other cell types to express the proteins that you are interested because they may be mutated in your disease. These are called heterologous expression systems that give you some cell complexity because there are now other proteins from the cell, but maybe they are not the proteins that are interesting from the cell that you want to study. Then we have animal models. Animal models reproducing or trying to model diseases in human diseases in animals covers a wide spectrum of cell complexity. You have a living organism, and depending on how that relates, it was mentioned earlier this morning, depending how the genetic complexity of that individual, of that organism, animal organism, relates to the one of your patient, and depending on how the cells that are interacting in this animal interact in the same way that they do in humans, you can reproduce or you can recapitulate something very close to the actual patient or something very far away. In the case of Parkinson's disease and many neurodegenerative diseases, we're here. We're in the lower end of how that relates to the actual patient. So we thought of using iPSCs. iPSCs are generated from patients. Then the genetic complexity will be the same. We capture the entire genome of the patient. The genetic complexity of whatever we see will be similar to that of the patient. And we are using one or a few cell types. So there is some cellular complexity, but far away from that of an actual living body. The general strategy in this case is, again, from a somatic cell from the patient, we can reprogram, generate iPS cells. These iPS cells will contain the entire genome of the patient and will be, in principle, devoid of epigenetic modifications. So if your disease has any genetic component predisposition to it, it will be also in the iPS cells, and these cells can be differentiated into any cell type. So we differentiate them into the cells that suffer this disease in particular, which is a specific type of dopaminergic neuron. And when we analyze these cells, if there was a genetic predisposition, these cells will get sick in the lab and we will be able to identify phenotype. This is the case, and we analyzed a number of years ago. There are two phenotypes that appear in this type of cells. So we generated iPS cells from healthy individuals and also from patients. Okay. Also from patients with Parkinson's disease, and we observed one early phenotype, which is the abnormal accumulation of alpha-synuclein in these dopaminergic neurons, okay? And a late phenotype that is the neurodegeneration. So these cells die upon prolonged culture or are more susceptible to die upon prolonged culture than control neurons do. So the first question that we can address with this type of models is this a consequence of the mutation that we see in these patients? And CRISPR-Cas technology, as it has been shown this morning as well, is a very easy technique to modify the genome of your cells. So we modify the genome of the iPS cells, and then the neurons that we derive from these cells are also modified. So we see that those from patients neurodegenerate and accumulate alpha-synuclein, those from controls don't do. So if it is a consequence of the genetic mutation, if we eliminate the genetic mutation, or introduce a genetic mutation in a control background, we should see if that was the cause. And it is the cause in these cases. So this is the accumulation of alpha-synuclein that we see in the patients, not in the controls. If we edit the mutation in this line, we see the accumulation of alpha-synuclein. If we remove the mutation from here, we see that the alpha-synuclein accumulation disappears. And the other phenotype that I mentioned, that was that these neurons in the control, in the patient, they die. If we remove the mutation from the patients, the neurons are just fine. So that will mean that if you get the mutation, you get the disease based on this model. But we know that this is not the case. We know that there are some relatives of these patients that share the mutation, they have inherited the mutation as well, but they never develop the disease. And this is also common for many other diseases. So there should be something, this is our hypothesis, that prevents, this type of individuals are called asymptomatic carriers or non-manifesting carriers. They carry the mutation but never develop the disease. So our first question was, will there iPS cells that carry the mutation show the disease that we develop in vitro? And if this is the case, if we remove the mutation, what happens? So in these two phenotypes, there are two very different behaviors. The accumulation of alpha-synucleins penetrates completely with the mutation. Those non-manifesting carriers, they show abnormal accumulation of alpha-synuclein. If we remove the mutation, this accumulation disappears. But the phenotype of neurodegeneration was something intermediate. See that we have three different individuals that we were able to trace and recruit for this study. And in all of them, in here, the length of the red bars shows the degree of neurodegeneration. The three non-manifesting carriers are the three bars to the right. You see that in all of them, there is more neurodegeneration than in the controls, the first two bars, but less neurodegeneration than the patients that actually show the disease. So there seems to be something that genetically protects these patients from neurodegeneration. And I'll go quick here, but the way we analyzed this was sequence the entire genome, well, the exome of these patients, try to identify something that could be associated to the phenotype of not manifesting the disease. And in one of the patients, we found one particular gene. This particular gene, this patient had a variant of the gene. It's not a mutation, it's a variant that about 2% of the population show. And this gene is particularly involved in organizing and function of the Golgi apparatus. So this is the Golgi here in green of a dopaminergic neuron from a healthy person that contains the variant in this gene that is normal in the population. 98% of the population contains this variant. This is a patient of Parkinson's disease with mutation in the LRRK2 gene, where you see that the Golgi is far away from the nucleus, protracted, and is not working well. This patient also has the normal variant of this gene. And this is the same patient. If we remove the LRRK2 mutation, Golgi goes back to normal, and the neuron doesn't die, as we have seen. In this individual that didn't manifest the disease, the Golgi was much larger and more active than in the normal population, the individual that contains this rare variant. And if we put this rare variant in the gene that has nothing to do with Parkinson's into the iPS cells from the patient, that prevents the Golgi from getting pyknotic and away from the nucleus, and has a normal Golgi function. And not only that, the more important, if you do that, then the neurodegeneration that we see in this patient that did develop the disease, you can see this in the bars, this is the number of neurons that die. If you put the rare variant, that goes back to almost nothing, and this is the individual that was protected, that has a low degree of neurodegeneration. If you remove the rare variant and put the common variant, the neurodegeneration goes up the roof. So we went then to the population of patients with Parkinson's disease. This is, as I said, something that occurs in 2% of the population. We gathered 500 patients, and as expected, 2% of them carried this rare variant. And in these patients, we saw that this is the age at which they develop the disease. And having this rare variant confers an almost 9 years advantage, or 9 years delay in suffering the disease. So there is a protection of 9 years for people carrying this variant to show the disease. Okay? So far, all of this has been in neurons. And as I say, if you remember the plot, the cell complexity, I mean, these are true neurons and the neurons that are of interest for these patients, but also other than neurons, there are other cells that can contribute to the pathology. And one of them is astrocytes. The good thing about the model in general, about this strategy, is that you can differentiate any cell type from iPS cells from the patient. So we differentiated also astrocytes from patients and from healthy controls, and we mixed cells and see if normal neurons on top of Parkinson's disease astrocytes were compromised, or if astrocytes from normal people could rescue dopaminergic neurons from patients. If you co-culture normal astrocytes with normal neurons, they grow just fine, they look like this. But if you take these neurons and culture on top of astrocytes from patients, they start suffering and showing signs of degeneration. In the most severe case, if you take neurons from patients and culture on top of astrocytes from patients, they undergo a very severe neurodegeneration that can be somehow rescued if you culture these Parkinson's neurons on top of normal astrocytes. And we found that one of the reasons by which this happens is that the astrocytes also accumulate abnormally alpha-synuclein. And in here, we introduce an endogenous tag into the alpha-synuclein gene of these astrocytes in a way that we can follow where this alpha-synuclein comes from. And we show that this alpha-synuclein produced by Parkinson's disease astrocytes is uptaken by normal neurons. So they are transferring alpha-synuclein, abnormal alpha-synuclein, to the neurons. Two cells, we're increasing the complexity, but if we want to increase more the complexity, and this is something that was alluded to and in detail this morning, we can grow this type of structures that we used to call them cell aggregates. Now we call them organoids, because everybody's calling them organoids. In this case, these are much dumber than the ones shown this morning. These are from ventral mesencephalic organoids, which is the equivalent to the structure that degenerates in patients. They grow over time. This is done in collaboration with Alison Mutri at UCSD, and this is after 20 or 40 days. You can see the complexity after 40 days. There are patches of dopaminergic neurons that crosstalk with other neurons. And after 90 or 120 days, 3-4 months, in this case, we can also see the appearance of astrocytes from the same iPS cells. So we can analyze the crosstalk of these two cell types within structures that are more reminiscent of what happens in the substantia nigra. And we can also use these organoids to colonize with microglia. These are green cells here, are microglia cells derived from the same iPS cells, so we can see how the microglia can influence this interaction. Finally, okay, perfect time. Well, also like to mention something that we have learned in the process. We didn't expect, but we were, if you go to any of these meetings of iPS cells, everybody's complaining that the cells that they generate from iPS cells are immature. And it is true, they represent late fetal, early prenatal cells at the best. But we realize that this can be also very convenient for modeling this type of diseases that develop with age. In the sense that the cells that we generate represent the cells that the patient had when he or she was born. And much, much earlier than any types of disease appeared in those patients. So there is a huge time window of opportunity to analyze phenotypes in those cells that we have never seen in patients. And this is one example of this. These are neuronal networks that we can organize with this in vitro, with these dopaminergic neurons and non-dopaminergic neurons from patients or control, mixed together, let them to form synapses to start working, and we can obtain electrical activity from these cells and see how this network is organized. And this is the structural network of a normal network, a network made with cells, neurons from healthy people. You see that this is a scale-free network, which is the sign of a robust and well-functioning network. In the case, if you do the same with a patient with this mutation that developed Parkinson, the structure of the network is completely abnormal. And you can see, if you can see here, if not, you can believe me, that there are on and off signs. Network fires all at once and gets silent, which is something that we never see in normal networks. So somehow, and these neurons are absolutely normal by all measures that we have done in them. They don't show any signs of neurodegeneration at this time point. And we can model this behavior. We can model the activity of dopaminergic neurons versus non-dopaminergic neurons, model mathematically this, and this is the results of the simulation. This is a simulation from a control network, where you can see that it's never off completely. And this is a simulation of a patient, a Parkinson's disease patient associated to LRRK2 mutation. You see that the behavior reproduces exactly what we see in the experiments, and we know what we have to twitch with in order to get this behavior, and we have checked, and this is the case. Again, the behavior, I will not call this sentient, by no means, but there is electrical activity that reproduces a specific pattern encoded in these cells. And we can go further and use something similar to what's shown. We call this, well, we used to call this injection of cells in the brain, something that we have been doing for many, many years. These are progenitors of these dopaminergic neurons injected in the neonatal rat brain, or mouse brain, sorry, in this case. And you let them develop in the mouse for 3 months. You see that some of them survive and establish synapses with other human neurons or with the neurons of the mouse host. And we are now analyzing the behavior of these networks. So I said we call this now hybrids, which also is fancier. So in essence, then we can use this technology to capture genetic predisposition to suffering disease and find phenotypes that are reminiscent of what you see in the patient and analyze why these phenotypes appear. We can see the molecular basis of these phenotypes, and we can also use, since these are cells that we can grow in unlimited amounts in the lab, we can use these cells and this type of strategy to screen for drugs that will revert these phenotypes. And if you're using phenotypes that predate the onset of degeneration, you can even find drugs that will prevent the appearance of the neurodegeneration. So we think that this has been proven. We are now in the way of using organoids in this phase of using organoids to increase the complexity, and even using hybrids to get to the complexity of the patient itself. I'd like to acknowledge all the people in the lab that has participated in these studies, and a stable collaboration with the lab of Antonella Concilio at the University of Barcelona, the funding, and of course, thank you for your attention.